and welcome back to the VGP Showcase. This first session for Series A, which is focused on vertebrates, we're going to hear about some new results coming out of the VGP project. That's the Vertebrate Genomes Project. And just to give a quick introduction, most of you heard about it in the previous session. This is a project to sequence the genomes of all vertebrate species at high quality to be used for understanding novel biological questions. And today we have a lineup of 5 speakers, and which, unlike in prior years, we're not going to focus so much on assembly this time around or annotations. We're going to focus on the new biological discoveries coming out of the genomes that we have been generating the last several years. So the first speaker up is Juan Kim and Byung-Jun Ko from Seoul National University, being advised by Hebal Kim, myself, and Arang Ray. And we're going to hear about some interesting things taking off in the last talk on what's happening with false gene losses and false gene gains in our assemblies. So go ahead and take it away.
Hello everyone, I'm Juan Kim from Seoul National University, and the title of today's presentation is False Gene Losses and Gains Revealed by VGP Assemblies. Before to start the presentation, thank you for giving me an opportunity to present today. And I would give a presentation about false gene loss part first, and Byung-Jun Ko would explain about false gene gain part next. So our team defined false gene losses as a gene with errors that might affect its own reading frame. And in order to classify, quantify, and characterize those false gene losses in previous assemblies, we aligned VGP and previous assemblies and projected VGP annotation on the previous assemblies for zebrafinch and hummingbird, platypus, and climbing birds. Among the 8 types of false gene losses we classified, today's presentation will focus on the missing genes and their characteristics. Here we show the ratio of genome-wide alignment between VGP and previous assemblies, and as you can see, from 4 to 12% of VGP genomic regions were not aligned with the previous one, which can be regarded as an approximation of previously missing ratio as well. Besides the difference of missing ratio among assemblies or among chromosomes, we also discovered that all species except climbing birds showed higher GC content in the not aligned region compared to that of aligned region. And it has been known that sequencing platforms such as Illumina or Sanger have trouble in sequencing this rich region. And considering climbing birds' homogeneous genome compared to that of other species, it seems that the previous assemblies of birds or mammal with rich genomic region have been missing those genomic parts due to the limitation of short read length sequencing platform in sequencing those rich sequences. And here we visualize the distribution of previously missing genes in zebrafinch VGP assembly. And panel A indicates the microchromosome and larger microchromosomes, and panel B indicates smaller microchromosomes and unplaced scaffolds. The percent in the outer circle indicates the ratio of missing genes, and the red bar indicates GISH-rich region, and the blue bar indicates the repeat-rich sequences. And we can see that in GISH-rich and repetitive smaller microchromosomes and unplaced scaffolds, we discovered a higher ratio of missing genes compared to larger microchromosomes or macrochromosomes. So it means that the distribution of missing genes was not uniform among scaffolds, but it was especially biased on GISH-rich scaffolds. Finally, we identified the effect of false sequence losses in GeneScape by comparing GC content and the ratio of unaligned region near protein coding genes for zebrafinch. We discovered a strong association between the mean of GC content and the mean ratio of unaligned region around genes, and especially GC-rich 5' upstream region and the 5' near exons were more frequently missing compared to other genomic parts. And interestingly, we found similar patterns in Anna's hummingbird and platypus, but climate perch shows missing ratio in a relatively uniform manner due to the homogeneous genome of climate perch. So this finding provided an overview of sequence losses around genes that can occur in a clade-specific manner, and also a symmetric pattern of sequence losses around genes imply that many of the previous genes have lost much of their G-rich 5' promoter or coding exon part. Here I finished my presentation on fast gene losses, and from now on, Hyungjun will proceed with the presentation on fast duplication and fast gene gain.
Hi everyone, this is Byung-Jun Ko. And in this presentation, we'll talk the fast gene gain caused by the type of specific assembly error, fast duplication. It is well known heterozygosity can make fast segmental duplication during assembly. The literal divergent sequence in same locus can be assembled in same chromosome or super scaffold or as a different scaffold. Then we can see the first duplication if these earlier will be mixed. So VGP assembly, which is the page assembly, and exclude the first duplication. So we align the VGP assemblies and the previous assemblies. VGP assembly accept alternate earlier sequences, then we can find first duplication for previous assemblies if there have these redundant sequences. So here is the amount of first duplication in previous Sanger or Illumina-based assemblies of zebrafinch, hummingbird, and platypus. We also classify the first duplication to heterotype and homotype duplication. The heterotype duplication is the first duplication made by heterozygosity, but homotype duplication is not. As a result, around 140 megabase pair were partially duplicated in zebrafinch's Sanger-based assembly, and around 30 and 40 megabase pair were partially duplicated in hummingbird and platypus previous assemblies as well. The crucial thing is that if the gene region were partially duplicated, they will annotate it as new gene region. This is first gene gain we talk. Here is first gene gain case in zebrafinch. The left LOC gene is a like gene of GBTB11, and we found this is a first gene gain. Here is detailed landscape of the lesion of GBTB11. We can see the first duplicated lesion we found has overlap with coding sequences of LOC and GBTB11. And we can see the first duplication lesion has a lot of homozygosity than the other lesions. This is typical characteristics of first duplication because the allele specific reads will be mapped to first duplicated lesion respectively. So the lead depths of first duplicated lesion will be decreased as well. Finally, the paired lead will be mapped to far away. The GBTB11 duplication clearly shows these traces of first duplication. So we found around 2,000 of first gene gain case in zebrafinch previous assembly, and 160 and 554 hummingbird and platypus as well. It is sure the other feature on the genome can be affected by first duplication. The zebrafinch LTR were reported as expansion than chicken, but they can be overestimated because first duplication can affect two gene family expansion. Actually, we found some LTR is the artifact made by first duplication. Then zebrafinch's LTR is just 1.8-fold higher than recent chicken genome. This is lower than the number of LTR previously reported. To sum it up, the first duplication can prevalently present in previous assemblies, and their impact will not be ignorable in downstream analysis. Thank you for paying attention. Thank you.
Thank you, Juan Kim and Byung-Jun Ko. Fantastic work and presentation. And I'd like to open up the floor for questions. So please type in your questions in the chat. And I'll take actually the first question. So I'm wondering, what do you, for Juan, what do you think it is as to why the smaller chromosomes have more genes and more missing genes in them as opposed to the bigger chromosomes?
Yeah, it is related to two factors, I guess. The first factor is the characteristic of bird genome, which have GC-rich microchromosome with the higher gene density. And since the high GC content is correlated with the higher missing rate, that's why the smaller chromosome shows higher number of missing genes compared to large chromosomes.
Okay. All right. So I want to make sure I don't see questions in the chat. Am I missing something, or no one has put a question forth yet? And while I wait for that answer, I'll ask Byung-Jun Ko, what is the prevalence of, in some of these older assemblies versus the new VGP assembly? Are most of the gene duplications large or small? Are they whole genes, partial exons, and so what? I mean, so forth.
Actually, I observed whole gene duplication in old assembly, and I also observed partial gene duplication as well. And versus VGP assembly and previous assembly, the previous assembly has a lot of first gene duplication than VGP duplication. So,
okay, I see. Yes, someone just asked that question. Okay, now the question's coming through. I'll ask one more. Let's see. Are most duplicates integrating multiple haplotypes into a single path? I think I know the answer to that is yes. At least you're getting duplicates put next to each other in the genome. All right. And we have time for one more. Are you able to assemble all copies of genes in your assembly? Is there one-to-one correspondence between read depth and gene presence for validation? In other words, are you using read depth to try to validate whether something is duplicate or not, a falsification?
Actually, as I know, read depth is prevalently used to validate varus duplication. Actually, the program of purge haplotic and purge dupe is already published, and they are based on the read depth to validate varus duplication. So read depth is very useful to validate varus duplication.
Okay. All right, great. So I'm going to have to stop there. There are lots more questions in the chat box that just showed up. I think I had a delay in seeing them, but you can go check them out later. And thank you very much for your presentation. So next up will be Chentao Yang from BGI, who works with Goji Zhang. And he's collaborating on a project, including with my lab, with Stephanie Marcus in my lab, on the marmoset genome, where we use a trio approach to actually assemble out the haplotypes. And we learned some new biology by separating out the haplotypes throughout the entire assembly. So Chentao, take it away.
Hello everyone, my name is Chentao Yang, and I'm going to give a quick talk about marmoset with the title of Evolutionary Insights from a Chromosome Level Diploid Genome Assembly of the Common Marmoset. I already talked about the background diploid genome assembly, heterozygosity spectrum between paternal and maternal genomes, evolutionary of new sex differentiation regions in marmoset. The common marmoset is a very small New World primate. It has been an important animal model for biomedical researches, such as neuroscience, stem cell biology, and immune studies. In this study, we applied a recently developed gene approach to produce a chromosome level fully haplotype resolved genome assembly for the common marmoset. The true binning approach needs a true sample. We sequence the illuminance shotgun reads for parents. Meanwhile, we sequence the peptide long reads, 10x genomics linked reads, banana, and high C reads from a male F1 marmoset. The right figure shows a VGP standard assembly pipeline, which I am not going to talk in detail, but in brief, the long reads need to be binned into two haplotypes by unique chimer sites from maternal and paternal shotgun reads. Then, two haplotypes will be assembled independently and after several rounds of polishing and scaffolding steps using 10x, Hi-C, and banana data, the final haplotype assemblies will come to the end. As a result, we assembled about 2.8 gigabits for maternal and 2.7 gigabits for paternal. They both have very high scaffold N50 and a very low gap ratio. In this figure, panel A shows a haplotype-specific chimer bubble plot of the marmoset assemblies. The size of each bubble indicates the total number of chimers counted in an individual scaffold, and the position of each bubble is plotted according to how many hapmers that maternal and paternal contain. As you can see, maternal and paternal hapmers are fitted very well. In panel B, we show the correlation between the assembled chromosome size and the chromosome length, estimated by the karyotype image data. In panel C, schematic plot mapping the assembled maternal and paternal assigned contiguous onto marmoset assembled chromosomes. Based on these high-quality assemblies, we got a chance to investigate the genetic differences between two haplotypes. In traditional genome sequencing efforts, heterozygosity was normally estimated by mapping the sequencing reads onto mosaic reference genome, resulting in limited phase information of the heterozygous variants. But our chromosome-level diploid assemblies allow us to directly compare the two alleles and identify the full spectrum of genetic variants, including single nucleotide variations, insertion, deletions, and other large structure variations. Here's a table to summarize the genetic variations we found. There are about 3.5 million SNVs, 232,000 small endiles, 6,000 large endiles, and 5,500 CNVs, and translocations, inversions. I'd like to highlight that the heterozygosity rate of our sequenced marmoset individuals is 10 times higher than estimations in most genetic studies generated using only SNVs. And we also estimated the sequencing error and the polishing error level. When it comes to the sex chromosomes, we identified 147 X-linked and about 6.5 megabase Y-linked sequences. It is about over 99% X-linked sequences present in single scaffold identified as X chromosome, where about 78 of Y-linked sequences in single scaffold identified as Y chromosomes. Based on this near complete X chromosome and Y chromosome, we can identify the precise boundary of PR and SDR. We found an inversion between human and marmoset PRs, which occurred specifically in the marmoset lineage by comparison with other primates' genomes. We also observed a long fragment of genomic sequence spanning 6 human PR orthologous and located downstream of these inversion regions in the X chromosome, which had become a new SDR in the marmoset. These translocations of marmoset-specific SDR also lead to the expansion of the male-specific region in the marmoset Y chromosome, making its PR the shortest among the primates recorded so far. By dating the formation of the marmoset-specific SDR, we found that the expansion is an young event. Okay, I want to summarize my talk, and first, we demonstrate the power of using trial binning approach to produce a diploid assembly with 2 alleles fully phased in chromosome level. Secondly, we show that this method can capture the full range of heterozygous variations and high accuracy rates between the 2 alleles in a de novo way. And thirdly, this method produces near complete sequences for both sex chromosomes, including the Y chromosome with highly dense repetitive elements. And finally, this method also provides opportunities to produce assemblies from species with extremely high heterozygosity rate. Finally, I would like to thank my supervisor, Professor Zhang Guojie, and my colleagues, and all people in VGP group, and Miko's teams from the Oxford University. And thanks to you for your listening.
Thank you, Chintao, for such a great presentation. We have 2 questions here, and I also want to alert to everybody and others to know that Chintao and also those you just heard from Korea a few minutes ago, they're staying up late here to answer these questions. And so please go ahead and send them in the chat box, as well as they'll be able to read them later on and answer them if we don't get to all of them today. So one from an anonymous, Chintao, is, how successful was annotations of complex genomic regions in these marmoset genomes, like the MHC genes or the NKC genes? How did you deal with haplotyping and chimerism in the marmoset?
So I think this the question is included 3 small questions about very complex genomic regions. I think we use a bin trial bin approach can produce a very long and continuous assemblies, and so we can get the very complete complete genomics regions like the MHC and other regions.
And what about chimerism? Did you check chimeras?
Yeah,
that's a chimeric.
We did check the chimerism, because we know the chimerism in the marmoset is very usually occurring. So, and we use SNV data to which generated from the 10x genomics data to estimate the chimerism level, and we estimate about 7 to 10% of the F1 marmoset, and we also check the region that's affected by this chimerism. That's very low percentage. So we think this chimerism is very which can be ignored by in our study.
Great. So basically, chimerism was there, detected, but it actually didn't affect the assembly. One question here is 4 votes. Is a new male region in the Y you discovered unique to marmosets or to Old World or all New World monkeys? The new Y region. Yeah, is the new Y region unique to marmosets, or is it present in all monkeys?
we discovered this Y monocyte specific male specific to Y region in the is just specific in the monocyte, because we
Okay.
also check his other new world primates. So it's just specific in the monocyte.
Okay, last question here from Peter Holland. Can you also use the trio reads to detect de novo mutation in the F1 generation, or is coverage too low for this use?
Actually, we use 10x genomes data to calculate the de novo mutations because we have a high risk depth, about 16. So we can use this data to detect the de novo mutations.
All right, fantastic. And I'll just say this last question on the sex chromosomes. It's based upon coverage for sex chromosomes. Okay, Chintao, great. You got some really good questions here. Thank you for staying up late there in China and being online. And please enjoy the rest of the conference. And so, and we'll some other questions might show up. They can email you later. Okay, next speaker. I'm introducing you to Nicholas Duxit. He's a postdoc who worked on the cockapod genome that the VGP group generated. And it's the first study coming out of this highly endangered species in New Zealand, where there are only a few hundred animals left on the planet. And he'll talk about deleterious mutations. So Nicholas, take it away.
Hi everyone, so today I'm going to talk about the genome-wide consequences of population decline in kakapo. So small populations are exposed to a range of threats. On one hand, we have external threats, such as habitat loss or overhunting. And on the other hand, we have internal threats, which include demographic processes and genetic processes, such as inbreeding, genetic drift, and loss of genetic diversity. So why does genetic matter? When population go through what we call population bottleneck, they will lose genetic diversity. And this genetic diversity actually represents the adaptive potential of species. So it's important to have as much genetic diversity to cope with future changes in the environment. The second reason why genetics matter in small population is that these small populations tend to accumulate mutational load, or the number of harmful or deleterious mutations. And this is because drift will overpower purifying selection. So these populations may have accumulate gene deletions or point mutation that may create stop codons within genes, meaning that fitness will decline in those individuals. And this process is referred to as genomic meltdown. Finally, in small populations, inbreeding will increase, and as a result, inbreeding depression or reduction in fitness will also decline. So in an outbred population, individuals may carry deleterious mutations, but these may be masked by a healthy variant, as you can see in this figure. However, as population becomes small, individuals may become more inbred, and there may be quite a few of those individuals that may carry both faulty variants or of a gene, meaning that the mutation will be expressed and the individual will suffer from inbreeding depression. So there's a clear relationship between increase in inbreeding through time and decline in fitness. And one example that illustrates the situation is that of the woolly mammoth. Woolly mammoth went extinct some 4,000 years ago, with the last population surviving on an isolated island, Wrangel Island. And two studies from a few years ago showed that a mammoth from the mainland from 40,000 years ago was much less inbred, or actually showed very little inbreeding compared to one of the last mammoth from Wrangel Island. Also, this mammoth from Wrangel Island showed a high number of deleterious mutation compared to that much older mainland mammoth. And this suggests that genetic factors may have contributed to mammoth extinction. So when it comes to small population, we have two different outcomes. On one hand, we have the one that I just talked about, a scenario of extinction driven in part by inbreeding and the accumulation of deleterious mutations. But there's an alternative scenario, which is referred to as purging of harmful mutation. And through a combination of inbreeding and peripheral selection, in some cases, some population can get rid of those mutations, can purge them. But one condition for this to happen is that inbreeding has to increase slowly, as opposed to a situation where a population goes through a very rapid population decline. So to test these two competing hypotheses, we studied kakapo, which is an endemic species to New Zealand, a flightless parrot, a bird that used to be widespread before human arrival. But by the 1980s, there were 50 to 60 birds left. There was one male, referred to as Richard Henry, surviving on the mainland, and a population of around 50 to 60 birds on Stewart Island. This male was used in the breeding program, and today the population of around 200 birds is managed on offshore islands. And we know also that these birds are highly inbred. So the first thing we did was that we mapped 14 historical genomes from the mainland, including that surviving male, and 35 genomes from Stewart Island. And to do that, we used a chromosome level assembly from the Vertebrate Genome Project. We estimated inbreeding and mutational load between those two populations. First, we reconstructed the past demography, and what we found is that as Stewart Island became isolated from the mainland some 50,000 years ago through after sea level rose, this population became gradually isolated, and we could estimate this divergence that was dating back to 9 to 14,000 years before present. So we have here a situation of a gradual isolation from the mainland and slow population decline on Stewart Island. Then we estimated inbreeding between those two populations using runs of homozygosity, which are large tracts on chromosomes that are homozygous, so identical between paternal and maternal chromosomes. And we found here a significant difference between mainland and Stewart Island individuals. And this difference was also significant when only considering fragments larger than 1 megabasis, which are supposed to indicate inbreeding that is recent, so that may have occurred in the last few generations as a result of human-induced decline. So this is not something completely new, but this time we had an accurate estimate of this inbreeding based on complete genome data. Then we estimated mutational load using two different approaches. The first one is referred to as GERP approach, which relies on conservation scores and relies on the identification of conserved regions among a large number of bird genomes. And prediction is that mutation arising within those conserved regions are likely to be deleterious. So we estimated this relative mutational load between in mainland and Stewart Island birds, and we found a significant difference between those two populations. And this purple triangle represents this sole mainland surviving male that was used in the breeding program. The second approach we used was based on 16,000 coding genes. So here we were looking at a SNP that were leading to stop codons, premature stop codons leading to breakdown in gene expression. And again, we found the same significant difference between mainland and Stewart Island birds. So taken together, our results show that inbreeding is higher on Stewart Island, that the mutation load is actually much lower on Stewart Island, which is consistent with purging. And it is possible that the long-term isolation and gradual isolation may have favored this purging. From a conservation perspective, it is quite possible that its mainland male and its offspring may have introduced deleterious variants in the current population. And because inbreeding is likely to remain high or even increase in the future, those harmful mutations may be expressed in the future. So it is very important here to balance the positive of genetic rescue, increasing genetic diversity, and negative effects such as increase in mutational load. So we would like to thank funders and the people who shared the data with us, and thanks a lot for your attention.
All right, Nick, thank you for that talk. And it was nice to hear of one of our first conservation projects coming out of the VGP effort and your efforts. So we have a question here from Anne McCarthy, something you and I know well about, which is, she says, I'm aware that there is a Tawanga species, that this is a Tawanga species, meaning a Maori species, and that the data may not actually be able to be shared internationally. How are the VGP dealing with cases like this? Go ahead and answer, and I can fill in for something that you might not know.
So the way I understand it is that this data has been, at least for the modern data, has been generated in New Zealand. So it was driven by New Zealand researchers with that Taonga perspective in mind. And the way I understand it is that this data is going to be stored in New Zealand. And then if researchers from overseas want to use it, well, of course, they're welcome to ask, but they need to ask a formal permission. And the idea or the condition is that these data shouldn't be used for commercial purposes. Then for the VGP, I guess you can understand, answer, Eric.
Yes, so yes, and what you just mentioned was for a lot of the resequencing data of lots of individuals. For the main reference VGP assembly, we did get special agreements on that particular animal called Jane, Kakapo Jane, and that is available publicly in the NCBI database, but all the other data is as you said. So, and we deal with these on a case-by-case examples to answer Anne's question. So from a question from an anonymous person, what is the mitochondrial and Y chromosome? What is the mitochondrial and Y chromosome diversity in the species? Is there a sex bias in extinction danger?
So we don't really know about the Y chromosome because we didn't look at it. One reason was that other people are planning on working on it. So we would, for this study, we only look at autosomes. But we published a paper in 2018, just looking at mitochondrial data. And that showed that there was very little. So there are just 2 haplotypes at the moment, and one that's overrepresented at 99%. So it's very, very low. And it seems to be nevertheless consistent with those results of a population that was naturally, so to speak, isolated over 10,000 years ago. And when it comes to sex bias in extinction, I'm not aware of any. Maybe there is a high risk at the juvenile stage. But I think the main issue from a genetics perspective is variance in reproductive success, where we are dealing with a lek breeding bird, where few males that are very successful will mate with lots of females. So the young males may not be able to breed for several years, if not decades. So that's, I think, the main issue at the moment.
Okay, unfortunately, we ran out of time, and you're getting lots of questions coming in. I'll just say something. Go look at it later. Somebody wants to know what is the benefit of having a high quality assembly for conservation purposes. Others, where's the Jane assembly come from, and so on. So Nick, these are all very good questions. Please look at the chat, see if you can respond to some of them, even in the chat window. Okay. All right. Thank you very much. So we'll have to move on. Our next speaker is Joanna Damas from Harrence Lewin's lab, a postdoc there, who use some of the VGP genomes and the broader G10K genomes for COVID-19 research, an important topic for our own species survival. Joanna, take it away.
Hi everyone, I'm Joanna. Today I'm going to talk to you about how we use comparative analysis of ACE2 proteins to predict the host range of SARS-CoV-2 invertebrates. And most of the work that I'm presenting here today was just published last month in PNAS. As you know, SARS-CoV-2 is the virus that is responsible for the COVID-19 disease, and in humans it uses the ACE2 protein as a cellular receptor to enter and infect the cells. There are 25 residues specifically in the ACE2 protein that were found to contact with the SARS-CoV-2 spike protein, and they determine the binding specificity. And under normal physiological circumstances, the ACE2 protein functions in the cardiovascular system and is important, for instance, to regulate blood pressure. So the ACE2 gene is present in all vertebrates. So our question was, what vertebrate species are potentially susceptible to SARS-CoV-2 infection through their ACE2 proteins? To try and answer this question, we obtained ACE2 sequence data from multiple sources, including the Vertebrate Genome Project, and our final dataset comprised ACE2 sequences for 410 vertebrate species that covered all of the major vertebrate groups and 87 vertebrate orders in total. We aligned all of these sequences and we obtained information, we extracted the information for the 25 residues that we know are important for binding in the humans. And our algorithm and predictions were mainly based with comparisons with the human binding residues, because we know that the SARS-CoV-2 is highly effective in using the ACE2 protein of humans to infect the cells. And here I have just to show you a subset of the species in our data set. And the white squares means that the amino acid for that species is similar, is identical to the human. And then we also have information of how conservative or non-conservative the substitutions are for each of the positions. So our prediction algorithm will classify ACE2 sequences in 5 different groups, from having a predicted very high to very low binding ability to SARS-CoV-2. And as I mentioned, it was primarily based in the identity to the human binding residues, but we also had some more specific rules in 8 different positions, because we knew that these residues were important to maintain bind stability of SARS-CoV, the previous SARS virus, and the ACE2 that it also uses as a cellular receptor, and that was the data that we had available when we were doing this work. We also took into consideration the colorings from the previous matrix. So which type of amino acid substitution was present in each of the positions. So we run this algorithm for our 410 species, and here I'm just highlighting some of the species in each of the groups that we classified. So in the very high group, we have 90 species, which includes the human, and the other 18 species were all old world primates and great apes, and they all shared the 25 residues with the human, so they were identical to the human in the binding residues. In the high group, we have a little bit more diversity. This group includes also primates, but also cervids, rodents, cetaceans, and the anteater. In the medium group, we have some species that are of agricultural importance, for instance, sheep, goats, and cattle, and we also have the felids. In the low group, we have, for instance, the dog and all its relatives, and some bats. From the 37 bats that we analyzed, 8 classified in the low group. The other 29 classified in the very low group, together with the pangolins, ferrets, and minks, and also all of the species that were not mammals, so fishes, amphibians, birds, and reptiles. We performed homology modeling to verify and to support our results. So the substitutions that were present in our data set were modeled and were classified as if they introduced unfavorable interactions in the binding domain or weakening interactions. As you can see from here, the results are highly consistent. With the number of weak interaction and unfavorable interactions, they increase as we move from the high group to the very low group. Our results are also highly in agreement with the existing infectivity data. And here I'm just presenting data from experiments in animals or in cells that were derived from the species that we are looking at, not experiments that had human cells expressing other species ACE2 proteins. As you can see from here, all the species in the very high category, they are infectable and they present severe symptoms, and the symptoms are similar to the human symptoms. And also all the species in the medium category, they are infectable. The only inconsistencies we had were the ferret and the mink, and we are at this moment investigating why this inconsistency is happening. So from our results, together with the infectivity data, we predict that the species that we classify in the very high and medium groups, they are potentially susceptible to SARS-CoV-2. So we advise that they should be focus of surveillance efforts. We also looked at the evolution of ACE2 across mammals. And the first thing we noticed that we can see from this plot is that the ACE2 protein, and here each of the panels, the y-axis represents the ACE2 protein from beginning to end. that the ACE2 protein in mammals is highly conserved. So conserved codons are marked with a green cross, and the codons that are under accelerated evolution are marked with the blue dot. So here from this panel, you can see that we have a highly conserved protein in mammals. But we also identified acceleration in some specific lineages, including the bats, carnivores, rodents, archaeodactyls, and cetaceans. And this acceleration is particularly high in chiropterans. When you compare it, for instance, with the primates, you can see that a higher number of blue dots, so codons under accelerated evolution, and we hypothesize that this is related with the higher level of interaction of bats with beta coronaviruses. This can also be seen in this figure here, where we have the structure of the ACE2 bound to the spike protein of SARS-CoV-2. And in blue, we have the residues that were found to be under accelerated evolution and also positive selection in the bats. There are 29 residues. And from these, 5 are overlapping with the bind domain, the red dots here. We also noticed that these are close or physically close to residues in the spike, in the SARS-CoV-2 spike protein that are under positive selection that are marked in green. So as a summary, our data can assist the identification of intermediate hosts and species that are potentially reservoirs and can also be used as models for SARS-CoV-2. So all the species that we categorize from the very high to medium categories, because they are potentially susceptible to SARS-CoV-2. We also found that there are multiple ACE2 residues that are important for binding that are evolving rapidly in several mammalian clades. And as I said, this is possibly due to their interaction with SARS-CoV-2 like beta coronaviruses. And at this moment, we are expanding this analysis to coding and encoding regions evolving in coronavirus infection pathways. So this work was a collaborative work involving people from many institutions and many countries, and I would like to thank them all for all their help and all of you for your attention. Thank you.
Hi, Joanna. Thank you for that wonderful presentation, and I'm glad to see that folks from the group are trying to contribute to our current pandemic. Let's see if we have any questions here. First person says nice work. All right.
Thank you.
So I'm curious. All right. So ACE2 is getting into some species, but not getting into species. Why are bats able to pick it up and they find the virus there, but their ACE2 receptor doesn't seem to be susceptible to picking up the virus?
So for the best of my knowledge, the SARS-CoV-2 virus was never found in bats. There's another virus that is related to SARS-CoV-2, but not exactly SARS-CoV-2. So that virus would be more adapted to use the bat's ACE2.
Ah, okay. All right, another question here from Anonymous. Have you identified any substitutions in the golden hamster or mink that can explain the inconsistencies between ACE2 predictions and infectivity?
We are looking into that now. So that's the next step. We are looking now that we have more infectivity data, because when we started our analysis, we only have 5 species with infectivity data available. So we were not able to use that information to improve our predictions and do something more based in that information. So now we are looking at the new infectivity data that exists for both species that can be infected by SARS-CoV-2 and those that cannot, so we can improve our predictions and do rules more related to specific residues that could be related with the species being able to be infected or not.
Okay, another one was ACE2, a single copy gene in all genomes?
Yes, it is single copy, yeah.
Okay, great. And one more. So animals living in seas will not be infected by the presence of the virus or its environment. Is that true?
Well, we hope it is true, but we also know that some cetaceans, they lack some genes related with immunity. So we need to surveil them more carefully and monitor them to make sure that they are not infected.
Okay, all right, Joanna, that's all the time we have questions for. So I wish we had more. There are two more there you can go look at. I just want to say that keep it going. We need your help. All right.
Thanks.
All right. So we're going to continue. We have one more talk. So we're going to go 5 minutes overtime for those who can stay. Before I introduce the next speaker, I just want to say you'll notice that all the speakers I've introduced here today are young graduate students or postdocs. They'll be looking for jobs. They're the future of our initiatives. So please reach out to them if you really like their work and want to hear more or want to consider them for a job. Last but not least is Ileana Bista, who's a postdoc with Richard Durbin's lab. And she's going to talk about using some of these VGP genomes to understand Antarctic fish genome evolution. And it's going to be live. Just give it 1 minute. We'll set this up 30 seconds and she'll be right on.
Okay, hello from me too. Today I'm going to talk about our work on the evolution of the Antarctic fish, the notothenioids, based on a new set of whole genome assemblies which we have generated. So basically, the notothenioids are the most dominant group of fish in the Antarctic Ocean and the Southern Ocean, and their evolution is very closely related to the isolation of the Antarctic continent from the surrounding lands, which caused a massive drop in water temperature and eradicated many other species at the time. Also, some key innovations, like for example, the appearance of antifreeze glycoproteins was also played an important role in the evolution of this group. So in order to study this fish better, we have generated a whole set of new genome assemblies, including some of the subantarctic species, going all the way up to the most derived group of the radiation, the ice fish. And then we have generated at least one PacBio assembly per major group and a whole other set of assemblies in between. And today I'm going to talk about some of the key results we have from analysis. Just wanted to start by introducing the Channel-Bull-Blenny assembly, which is actually a reference assembly we generated in collaboration with the Vertebrate Genomes Project. And the Cotoberca gobio, as the species name, is the order representative of the perciforms for the VGP. It's a high quality reference assembly assigned to 24 chromosomes and is generated according to the VGP quality standards. So in order to compare this assembly, we ran synthetic analysis against the high quality reference of the Japanese Bedaka. And we can see that despite the fact that these two species are separated by almost 200 million years of divergence, we still don't find any major interchromosomal rearrangements between the two. So we have nevertheless identified a 65% genome expansion within a species of the radiation, which is driven by activity of transposable elements. Here we can see that there's multiple types of transposons which are actually contributed to this expansion. And we can also see through divergence analysis that there's been a major burst of newly diverging Ts within the Antarctic clade, as you can see here in the repeat landscape analysis in comparison to the subantarctic basal species. Additionally, we have been working on reconstructing the antifreeze gene locus for this species. As I mentioned at the beginning, the antifreeze genes were a really key innovation that enabled the notothenioids to avoid freezing at water temperatures below zero, as it happens in the Southern Ocean. Nevertheless, reconstructing and assembling correctly this locus had been very difficult in the past because of its repetitive nature and structure. Here, using two new chromosomal assemblies, we have reconstructed the derived locus on the South Georgia icefish genome, and also the ancestral locus in the Channel-Bull-Blenny assembly, which is located on chromosome 16. And this is actually the point where the FGP gene expanded. In the South Georgia icefish, we have identified 15 copies of the FGP genes on an entire locus that spans almost 1 megabase in length. Additionally, we have been working on studying the loss of hemoglobins within the Canichthidae icefish family. The icefish are actually the only known vertebrate which are completely lacking functional hemoglobins. That makes their blood look white, which is really surprising since they don't have any erythrocytes, and this is something that cannot be seen in any other species anywhere else. So basically, we used 5 new pack bioassemblies we have generated and a number of other available good quality assemblies to reconstruct the 2 hemoglobin loci, the LA and MN loci. As you can see here, we have confirmed the complete loss of hemoglobin genes in the MN cluster, while we can see only a remnant of a gene, a pseudogenized copy of the A globins in the ALA cluster. Additionally, we have identified multiple species-specific cis duplications within other species. And just to finish, I would like to thank very much the VGP Consortium for their input in this work, our group at Richard Durbin's and Eric Miska's lab in Cambridge, and our team at the Sanger, as well as all my collaborators who have been contributing to this work. Thank you very much. I also have a poster in the comparative genomics session for more information. Thank you.
Thank you, Ileana. Very nice. And because we're out of 7 minutes overtime, and I don't see a question yet, although there's a delay on the questions, what I am going to encourage you to do is go see Ileana at her poster. But I can say her work clearly shows that the higher quality assemblies are necessary to look at the duplicated gene families here that are very interesting. So thank you for that. And thank you for the whole group of new and up and coming young scientists working on these high quality genomes and generating them. And thank you for your attention. I encourage you to go to the poster sessions, and next we're going to see what, and whatever is next on your schedule that you like to go see. Thank you very much.